Hello guys, welcome to Dr. Science. So today we are going to talk about the proteins of a T cell receptor and the importance of the T cell receptor in MHC restriction. Okay, so first of all I would start with the basics. So basically we are having adaptive immunity, and this adaptive immunity is mainly divided into two types, which is your humoral immunity and the other one is your cell mediated immunity. So when we talk about humoral immunity, I will discuss this humoral immunity in detail in the future lectures. So when we talk about humoral immunity, basically the B cells will produce antibodies. Okay, this this is called as your humoral immunity. As you know, antibodies are your sticky glycoproteins. Antibodies are your sticky glycoproteins, and these sticky glycoproteins can go and attach with microbes. Suppose this is a microbe here, so these glycoproteins can go and attach with these microbes. Okay, so that antibodies or the sticky glycoproteins are produced by your B cells. And why they are named as B cells? Because they are mainly derived from bone marrow. B cells are derived and matured from bone marrow, hence they are called as your B cells. Now today the topic is about cell mediated immunity. When we talk about cell mediated immunity, we are mainly talking about your T cells. Okay, T cells, why they are named as T cells? Because T cells are derived from your bone marrow. Okay, they are derived from your bone marrow, and from bone marrow they will travel into the blood, and from the blood they go to an organ called as your lymphoid organ. Okay, let me clear this one. So basically suppose this is the sternum. Behind the sternum there are lymphoid organ. Okay, this lymphoid organ is called as your thymus. What is this lymphoid organ called as? This lymphoid organ is called as your thymus. So basically T cells are produced in the bone marrow, they travel into the blood, and from the blood they go to the a lymphoid organ which is present retrosternally called as thymus, and in the thymus they get matured, hence they are called as your T cells. So basically when we are talking about T cells, there are mainly three types of T cells. Suppose this is a T cell, and here is also a T cell, and this is also a T cell. The first T cell we are going to talk about is your T helper cells. Why they are called as T helper cells? Basically T helper cells the T helper cells will produce some cytokines such as interleukin 4, 5 and 13. I will discuss about these in the future videos, but you have to trust me. So basically the T helper cells will produce some cytokines like interleukin 4, 5 and 13, and when these interleukins are produced, the B cells will start converting into plasma cells, and once they are converted into plasma cells, they produce sticky glycoproteins called your antibodies. So basically T helper cells are helping in the conversion of B cells into plasma cells. Okay, so basically when we are talking about plasma cells, this is the structure of a plasma cell, and it is having a peripheral cartwheel shaped nucleus. Okay, anyways, I will talk about B cells in my future lecture. So first we talked about T helper cells. Next, the other type is your T cytotoxics. Why it is called as T cytotoxic cells? So basically this T cytotoxic cell is very dangerous. Why it is dangerous? Because suppose this is our cell, and inside the cell we are having, suppose, a virus is there inside our cell. So this T cytotoxic cell can kill this intracellular virus. Okay, it can kill the intracellular virus or bacteria. Hence, it is more potent compared to any other T cell. Hence, hence cytotoxic. Cytotoxic because cyto means cells. It is toxic to the infected cells. Hence, it is called as a T cytotoxic cell. Now, finally, we are going to talk about your T regulatory cells. Now, imagine there is a virus, and this virus is producing, due to this virus there are lot of T cytotoxic cells. Okay, there are lot of T cytotoxic cells, and there is rapid deletion or rapid apoptosis of your cells. Now you have to control these cells from dying. If there is too much activation of your immune system, they will not only kill your virus infected cells, they will also kill your normal cells also. So what God has given is T regulatory type of cells. These T regulatory types of cells will tell the T cytotoxic cells to calm down. They are regulating, or they are calm, they are letting the T cytotoxic cells to calm down, and thus helping in decreasing in T cytotoxic activity. Okay, that is the function of your T regulatory cells. Now I hope you guys have understand everything about it. So basically your cell mediated immunity consists of mainly three types of T cells. One is T helper cells, T cytotoxic cells, and T regulatory cells. Okay, so now we move on to the next topic, which is your T cell receptor. Suppose this is a T cell. Now this T cell, either it is a T helper or T regulatory or T cytotoxic cell, obviously it is going to contain a receptor. Now this receptor here is called as your T cell receptor. Now what is the T cell receptor is made of? That is, what are the proteins involved in this T cell receptors, we are going to see. Okay, and I forgot to tell you that T regulatory cells also help in prevention or limit immune response against self antigen. So what is the meaning of this? Suppose there is a T cell. This T cell, it is attacking our heart cells. Suppose this is our heart cells, or heart is having some antigens, and I will name this antigen as X antigen. Okay, now these T cells are going and activating the T cytotoxic cells are going and deleting this X antigen. Now that can be called as your autoimmunity. In autoimmunity, the T cells will go and attack our own antigens, which can be fatal. Okay, so what the T regulatory cells will do? The T regulatory cells will tell the T cytotoxic cells not to attack our own cells. Hence, it will decrease the autoimmunity component. So basically, T regulator cells are helpful in limiting the immune response against self antigen. So I already told you the T cells, whether it is T helper, T regulatory, or T cytotoxic, can have a T cell receptors, and now we are going to see what are the proteins present on your T cell receptor. So this is, suppose if we think this is a plasma membrane of a T cell receptor. Okay, let me draw this one. So this is the plasma membrane of a T cell. Okay, now this obviously it is going to have a T cell receptor, but T cell receptor is not like this. Okay, the T cell receptor usually consists of a heterodimer because it is going to consist of a heterodimer of alpha component and a beta component linked together with disulfide bonds. So basically a T cell receptor, so this is your T cell receptor, and it is a heterodimer because alpha and beta, they are heterodimers, right? Dimers. It consists of alpha beta heterodimer linked with disulfide bonds. This is the basic composition of a T cell receptor. Now this alpha and beta proteins which are present on the T cell receptor not only consist of alpha and beta but also consist of other polypeptide chains such as your delta and epsilon. Okay, now this delta and epsilon polypeptide chain is called as your CD3 proteins. Okay, so this delta and epsilon polypeptide chains are called as your CD3 proteins, and these are linked non-covalently. Okay, and on the other side we are going to again have a gamma. Okay, this time we are going to have a gamma and epsilon. This is also gamma and epsilon polypeptide chain, which is also named as your CD3 proteins. And aside from that, you also contain a polypeptide chain name as your Z. Okay, so this is the symbol like this. This is called as your Z polypeptide chain. So this Z polypeptide chain is named as your zeta. Okay, so it is called as your zeta. So basically, so this is the T cell receptor. Let me draw this one clearly. So basically this is the T cell receptor, and T cell receptor is consisting of CD3 proteins, which consist of mainly alpha, delta and epsilon chain, and gamma and epsilon chain, whereas it also consists of zeta chain, which is zeta protein, which is consisting of your two Z polypeptide chains. Okay, so basically this is the composition of your T cell receptor. So this entire thing is called as your T cell receptor. Okay, so this alpha and beta are called as your T cell receptor, whereas these three down here called the zeta CD3 proteins, they are same in all T cells. Okay, in all T cells like T regulatory cells, T cytotoxic cells and T helper cells. Okay, these are same in all T cells like T regulatory, T cytotoxics and T helper cells. Whereas the alpha beta component, the alpha and the beta component, they may 95% of the T cells are consisting of alpha beta heterodimers. Okay, the remaining 5% can consist of your delta gamma composition, delta and gamma composition like this. Delta linked with disulfide bonds and gamma composition. It is present in 5% of T cells. I will explain this one. Wait for a minute. So basically this T cell receptor T cell receptor consisting of alpha beta, and together with CD3 proteins, I will not draw entire thing, the CD3 proteins and the zeta chain like this. This entire complex is called as your T cell receptor complex. The entire thing is called as your T cell receptor complex. Now I would like to say a little about MHC restriction. I think you guys have heard about MHC. MHC means major histocompatibility. Major histocompatibility. This thing is very important, but I would like to say a little bit about it, and in the future lecture I will do a complete lecture on MHC compatibility. So suppose this is a T cell here. Now this T cell is going to consist of a T cell receptor. Aside from T cell receptor, there is also another receptor. This receptor is called as your CD4 or CD8 receptor, or your CD4 or CD8 proteins, I will call them. Okay. Let it be receptors, okay, for the sake of this lecture. So basically the CD4 and CD8 receptor, what is the function of this CD4 or CD8 receptor? So suppose if there is a virus, or we will take a bacteria, and this bacteria is having certain antigens. I will name this antigen as M proteins. Now these M proteins, this bacteria should get killed, right? Because it is harmful to our body. So what the dendritic cells will do, there is a cell called as your dendritic cell, and this dendritic cell will go and take a bite of this. Okay, once it will take a bite of this M protein, it will display the M proteins on its surface. It will take a bite of the M proteins and display the M protein on its surface, and this antigen, this M protein is called as your antigen, right? This antigen is presented to the T cell. So basically the dendritic cell is going and taking a bite of the M protein, and once it take a bite of the M protein, the dendritic cell will go and present this antigen to the T cells, okay, because T cells will ultimately go and kill the bacteria. So basically the dendritic cell is called as your antigen presenting cell. It is called as your antigen presenting cell. Why it is called as your antigen presenting cell? It is taking the antigen M proteins and it is digesting the antigen inside its body and displaying it on the surface of its cell, okay. So in order to present this antigen to T cell, it should contain a special type of receptor, and this receptor here is called as your MHC receptor. So basically, once again, this is the T cell. So T cell is going to contain T cell receptor, and not only that, they are going to contain CD4 or CD8 receptor. So on a fine day you are getting infected by a bacteria, and it is going to have an antigen called as your M protein, and it has infected your body. So what will happen? Your body will produce a cell called as your dendritic cell, which will go and take a bite of your M protein, and once it takes a bite of your M protein, it gets digested inside the dendritic cell, and once it is digested, it is going to get displayed on the surface. It digests the M proteins or the antigen, and once it is digested, the peptides are shown on the surface of the cell by the help of a receptor called as your MHC. It can be type 1 or type 2. I will discuss this one in the future lectures. So basically the dendritic cell will go and present this to the T cell, to the T cells. So there is shaking of hands there is shaking of hands. This antigen is presented to your T cell. Okay, so this type is called as your MHC restriction. What is MHC restriction? MHC restriction is nothing but not everything can present to the T cells. Okay, like neutrophils and macrophages, they cannot come and display this antigen to the T cell because it will be like a clutter. Imagine there are these Ukrainian people, right? What happens if the Ukrainian people will enter Poland, each and every one of them? Obviously, the people present in Poland and the Ukrainian people will going to have a lot of fight and clutter, right? So, what God has done is that the people who, the cells which have only MHC receptors present on them. So basically, suppose this is a T cell and this is a T cell receptor, and T cell receptor is also having a receptor called as your CD4 or CD8 positive receptor, and here is a antigen presenting cell, and this antigen presenting cell is having MHC receptor. So basically, not everyone can react with the T cell. Okay, only special people can contact the T cells, and those special uh, type of cells are nothing but the cells which have MHC receptors present on them. Okay, so MHC receptors are like hands, and these hands are having a antigen present on their surface, and these hands will react only with T cells, and this is called your MHC restriction. Okay, I hope you understand the concept of MHC restriction. So basically, I already told you that not everyone can react with T cells. That is the important point to remember. Only special type of dendritic cell or antigen presenting cells which have MHC receptor present on them can go and react with the T cell. Okay, that is called as your MHC restriction. I hope you understand that one. But I already told you that 95% of the T cell receptor consist of alpha linked with beta heterodimer, and 5% of the T cell receptor will consist of delta or gamma composition, delta gamma heterodimer. So basically, suppose this is a T cell membrane, and this T cell membrane is going to consist of delta linked by disulfide bonds together with gamma composition. This is called a delta gamma heterodimer. Now this delta gamma heterodimer, obviously they don't have MHC restriction. This is very important. I already told you this MHC restriction is present in 95% of alpha beta heterodimer, whereas 5% T cells which are consisting of delta gamma heterodimer on their T cell receptor, they don't have MHC restriction. It means every cell can react with these T cells which are having this delta gamma heterodimer, and these type are mainly present in the urogenital system or beneath your skin or on your mucosal surfaces. Suppose if there is suppose this is your skin, and one day you are running and you fell down, the skin will get damaged, then the microbes will get entered. Okay, now in order to kill these microbes effectively, if there is MHC restriction, T cells will not react with anyone except MHC receptors, right? So obviously there will be a delay in the quarantine of the microbe. So hence the T cells with the T cells with delta and gamma receptors are can be easily delta and gamma receptors can easily quarantine the microbe. Okay, that is the importance of this one. Now basically, suppose this is a T cell. I already told you T cell is going to consist of a T cell receptor, and not only that, it is going to have a CD4. For this instant, I will take a CD4 positive T helper cell. Okay, we are going to take a T helper cell which is having a CD4 receptor. So it will react with antigen presenting cell. Suppose this is an antigen presenting cell, and this antigen presenting cell will only have MHC class 2. Okay, it means the T helper cell will only react with anti T helper cell which is consisting of CD4 positive receptor will only react with antigen presenting cells which will have MHC class 2 receptor. Okay, whereas in the case of a T cytotoxic cell, this is a T cytotoxic cell. This T cytotoxic cell is going to have a receptor called as your T cell receptor. On the top of that, it is going to have your CD8 receptor. Okay, in T helper cell we are seeing CD4 receptor, whereas in the case of T cytotoxic cells we are going to see CD8 receptor. So what will happen? Suppose this is an antigen presenting cell. Now here the receptor is different. The receptor is mainly MHC class 1. Again, the CD4 positive T cells will react with only antigen presenting cells with MHC type 2 receptors, whereas T cytotoxic cells with CD8 receptor will only react with antigen presenting cell with MHC class 1 receptor. Okay, so now I am going to show image which is taken from your pathology textbook. So as you can see here, basically the T cell receptor is going to consist of your alpha and beta heterodimer linked with disulfur bonds, and this is called your T cell receptor. And as you can see here, the CD3 proteins consist of a delta and epsilon chain, polypeptide chain, and the CD3 proteins here is consist of gamma and epsilon polypeptide chains. And here you can see the 2Z polypeptide chains consist making your zeta chain. Okay, now this entire complex is your T cell receptor complex. Now as you can see here, not only there is a T cell receptor, this entire thing is a T cell receptor, it is also consisting of your CD4 positive receptor. Now I am going to ask a question. CD4 positive T cell reacts with, yes, CD4 will react with MHC type 2 antigen presenting cell. So basically, suppose this is the antigen presenting cell and this is the plasma membrane of the antigen presenting cell. It is going to react with your class 2 MHC molecule. Okay, so antigen presenting cells will have class 2 receptors, MHC type 2 receptors present on them. Now what about CD80 and CD86? This one I will discuss in the next video. So thanks for the understanding.